Okay, this is your chapter 16 notes on DNA and DNA replication. I apologize for my voice. Um, it's pretty late at night, at least for me. Here, but always like it's early. Yep, only 10 o'clock. All right, let's see. Where's my, here we go. All right, DNA is the genetic material. Um, we didn't always know that DNA is what made us who we are. We didn't know that DNA passed from parent to offspring. It took a lot of years of research to figure that out. The role of DNA in heredity stemmed from work that scientists did with bacteria and also viruses. Remember, viruses are not alive, but one of the defining characteristics is they contain either DNA or RNA, and they can pass that along to living cells. We'll start with each of these individuals as part of your reading for this chapter. You're going to read about the background and the history of some of these guys. To the work of, oh wait, no, I think I cut that out this year. Okay, so I'm going to cover each of these individuals in this PowerPoint, and I think that you have to cover them in one of the Bojan videos. I think I cut out that as a reading. You're reading it different this year. Okay, so Griffith, Frederick Griffith. So what he did is he studied bacteria that cause pneumonia. There are two strains. One was pathogenic, which means that it caused disease, and one did not. It did not cause any harmful effects. When he killed the one that caused disease with heat, he just heated it up, and he mixed it with cells. He found that there are cells that were living with non-pathogenic, that some strains of the living became pathogenic. So they changed, they transformed. They call this like a transforming principle or something. And this new pathogenicness was then inherited by future descendant bacteria cells. So let's take a little picture. So this is basically what he did. And he injected them into mice, and the pathogenic ones would kill the mice. The ones that were pathogenic, but he had killed them, or killed the bacteria, they would remain healthy. Sorry. But if he had killed the pathogenic, but he'd also put in the living cells, then he found that the mouse still died. So he knew that those cells were picking up something. The other evidence came from bacteriophages, which are viruses that specifically infect bacteria. Hershey and Chase found that DNA from the virus was injected into bacteria, which caused the bacteria cells to make new copies of the virus. And they did a little experiment, and this is actually explained a little bit better in your book. But the color here is radioactive. That they basically identified the protein around virus, and they labeled it with something radioactive. And when they did that, they noticed that the radioactivity stayed outside of the cell. But when they labeled the DNA with the radioactivity here, then the radioactivity was actually identifiable inside the cell. So they knew from this, basically, that it wasn't the proteins of the virus that were being injected into the cells, it was the DNA. That was a really, really big step in understanding what was being passed from one generation to the next. Nope. Okay. So DNA structure. You need to just know the DNA structure. We covered this in Bio 1, but we're not going to spend a lot of time on it in AP because it's really memorizing. You have to memorize how DNA is structured. So DNA is made up of building blocks called nucleotides. Each nucleotide consists of one nitrogen base, a sugar, in this case for DNA, the deoxyribose, and a phosphate. Sargass has some rules that talk about the base pairing, these nitrogen bases, and how they're paired in DNA. First of all, we have four bases, A, T, G, and C. A stands for adenine, T stands for thymine, G for guanine, and C cytosine. And how much of each of these you have varies depending on what species you are. And it's about the same, like all humans have about the same. As long as you're a human, you're going to have about the same. Really, our DNA is more alike than it is different. Um, Within a species, the number of A should equal the number of E's, and the C should equal the number of G's. G. This was one piece of evidence that made scientists understand that A is always going to pair with T, and G is always going to pair with C. That is complementary basis. That was one piece that identified that. So here's one side of DNA. This is what we call the backbone. It consists of a phosphate group, a sugar, phosphate group, sugar, phosphate group, sugar, and so on and so on. And that's the backbone of the back of the ladder. And then these are the nitrogen bases. And you'll notice, when we talk about this in a minute, that two of these bases are one ring, and then two of them have two rings. Okay, and that's really important a little bit later. This piece here is what I was talking about as being a nucleotide. One phosphate, one sugar, one base. So that's a nucleotide. And basically DNA is just made up of several nucleotides all put together. So how do we know about the structure of DNA? Because actually we cannot see this at the molecular level. We can, however, use electron microscopes to see the electrons. I know that sounds kind of weird. I'm not really sure I fully understand it because I've never looked at an electron microscope in my life. I just know that scientists know this, but they can't see it per se. Long ago, there was something called X-ray crystallography, and this is Rosalind Franklin. She's one of the females that is a big spearhead in DNA research, and she was the first to kind of confirm that DNA was a double helix, which is what Watson and Crick actually assumed was the structure of DNA. She confirmed that using X-ray crystallography. Franklin's picture further noted the DNA structure is uniform in diameter. We'll talk about that in a second. In order for that to be the case, one purine must fit with one pyrimidine. Remember when I said a few slides ago that two of these have two rings, and two of them have one ring. The ones that have two rings are purines. The ones that have one ring are pyrimidines. And I remember this because two rings are larger, but the word is smaller, and one ring is smaller, but the word is longer. That's the best way I come up with things to remember it. Add that to what Sargraph said about the amount of A being equal to the amount of T, and the amount of T being equal to the amount of G, and you realize that A always has to go to C, and C always has to go to G. And so, pyrimidines were paired together to be 2Y at certain points, and then the pyrimidines as they paired would be 2 men. DNA wouldn't be uniform in its size. The only way for that to be the case is if one and one pyrimidine pairs together every time. And that was consistent with the X-ray data. And then one thing I'm going to note here is right here is where these two bases bond. This is a weak hydrogen bond. And that's good that it's weak because DNA has to split apart to replicate in all sorts of proteins. The other thing about DNA is it's parallel. That means one strand. That means that one strand runs 5' to 3', and the other 3' to 5'. But what does 3' and 5' actually mean? What that means is that if you actually count these sugars up, the way to do this is these are 5 sugars, 1, 2, 3, 4, and 5. This phosphate is attached to the 5th sugar in the sequence. And so they call that the 5' end. And then over here, we do the same thing, 1, 2, 1, 2, 3. It's actually right here. This would be, oh wait, sorry, it goes wrong. 1, 2, 3. I guess that you count it that way, but this is the 3', so third carbon in here. Oh, what happened? And so that's why it is. Now up here, it's just the opposite. You'll notice that the way that it's oriented is different. This side is one direction, and the site is going the other direction. It's called anti-parallel, meaning that they're parallel, but they run in opposite directions, is what that means. And then these are just different ways of looking at DNA. This just looks more at understanding a basic structure. This gives you more of a chemical formula and shows you the atoms, and this shows you what it would actually look like with the atoms. It's called a space filling model. Okay, the next topic we're going to talk about is DNA replication. How do we make copies of our DNA? If you remember in the cell cycle, 90% of the cell's life is spent in interphase. Here. And then there's G1, S, and G2. S phase is when DNA replication happens. It has to happen before a cell divides from one cell into two cells, because if you have 46 chromosomes in this cell, you want each of these cells to have 46, which means that at some point you have to multiply that DNA by 2. And when we do that, if you start with 46 here, trouble tonight, 46, we go to 92, and then we go from there and split into the two 46s. So how does this process happen? That's what we're concentrating on in this particular chapter. One thing is that it's semi-conservative, meaning that when DNA replicates, each of the two daughter strands will have one old strand and one new strand. A long time ago, there were actually 3 models that were proposed by Messelson and Saul, and they did some experiments, and I can describe these experiments if we can, that supported that it was a semi-conservative model. So what that means is, here's the parent molecule of DNA. It separates, and then each new strand has an old strand of DNA and a new strand that has been newly synthesized. And so these were the models that were proposed. One was that the whole DNA model, double helix versus copy. And then semi-conservative, which is what we now know it is that one old strand existed and then one new strand. And then dispersed would be like little bits of old, little bits of new, little bits of old. And so it was really broken up. Both of these would not be correct. DNA makes copies of itself with remarkable speed and accuracy. So to kind of put this in this perspective for you, if we were to print all of the base pairs in your cells in the type font of your textbook, the 6 million nucleotide pairs in a diploid human cell would fill about 1,200 textbooks. That's how many letters are in your DNA. The size of yours. Yet it takes only hours for your cells to copy all of the DNA in a cell. And you can't imagine taking 1,200 of your textbooks and copying every single letter down. It would take you a whole lot longer than a couple of hours. We're going to do most of the notes about what happens during DNA replication in pictures. So add notes as we go, write things down that I say, make notes of what things are. We're going to model this in class. Okay? And we're going to like get out pieces of paper and we're going to cut things apart. You're going to have to physically show me how this happens. So in bacteria, this is an example of E. coli bacteria. Bacteria is round, or DNA is round in most bacteria. And so DNA replication is actually very simple. It starts with creating a bubble, or what we call a replication fork, right here. And then DNA copies itself in one direction and in the other direction until it's finished. And the reason it goes in both directions is it's faster. And so it's really pretty easy. It copies, and once it's done, you have 2 copies of DNA. In a eukaryotic cell, because DNA is linear, it doesn't work quite as well. If you start on one end, it's just going to go in one direction. So you have to have multiple replication forks or replication bubbles, and each of these are going to go in both directions, synthesizing, and then it'll kind of fill in these gaps here. And so as that happens, that makes it go a little bit faster. So it's not just putting one and going in one direction. Here's a quick picture that highlights some of the enzymes that are important in DNA replication. Helicase is what unwinds the DNA. The DNA is a twisted lag, it's a double helix. It's going to unwind that helix so that it's not twisted anymore. When that happens, as it's happening, topoisomerase is going to relieve the tension or the strength. As DNA is unwinding, if you could think about maybe like unwinding a piece of yarn, or if you do like a friendship bracelet that's braided and you're unwinding it, it's going to create tension behind where it's unwinding. And so topoisomerase is going to relieve that tension. Primase is going to add RNA primers, which are just bases right here paired up on the DNA. And there's a little bit difference between DNA and RNA, and that's going to be the last slide in this lecture. And then these single-stranded binding proteins are just going to hold everything in place. So here's our parental DNA. It's going to unzip, and then we're going to concentrate on one side at a time, but both sides are going to get copied. So we'll start with this side of the DNA here, up here. So here, it's going to unwind on the 3' end, okay? And DNA is always synthesized 5' to 3', always, okay? That's going to make a big difference later on. So a primer is going to come in, and it's going to add bases here, okay? And then it's going to start synthesizing DNA. Eventually, those primers will get replaced with DNA, okay? And it's just going to synthesize in this direction. So this keeps opening up, it keeps going in this direction. It's very easy to keep synthesizing, okay? And you can see in this picture how it's continuing to do that. Always going to synthesize 5' to 3'. Well, what's interesting is, on the other end, here, if we start synthesizing 5' to 3', what about back here? So we're going to keep going. But at some point when we start synthesizing this way and we're unzipping back here, we're going to have pieces that have to be resynthesized. So you end up starting a primer, going, starting a primer, going, and then you have to backtrack, is basically. I'll show you a better picture of that in a little bit. You have to backtrack on this strand, and then you have to fill in these gaps here. And these gaps are called Okazaki fragments. So you can kind of see it here. If it's unzipping this way, and DNA is synthesized this way, every time you open this up here, you're going to have to restart a primer, and it's going to go that way. So this is what we call a lagging strand, and the one that's synthesized continuously is called the leading strand. So you can see right here, this is the leading strand, this is the lagging. Wherever this bubble started, it's just going to be the opposite when it goes in this direction. This is going to be leading, this is going to be lagging. And in the lagging strand, you end up with Okazaki fragments that they have to then put back together or seal. So you're going to have a lot more RNA primers on that end. So I talked about these enzymes, but I just wanted to list them here for you so you have them. Helicase unwinds, topoisomerase release strain, primase starts the primers, DNA polymerase is what actually adds the nucleotides to a pre-existing strain. So DNA polymerase is what's going to add these blue pieces here. DNA polymerase can add nucleotides only to the free 3' end, and it always synthesizes 5' to 3'. That means that one strand can be continuous, like I showed you, and the other is not. That's called the lagging strand. It's synthesized in a series of segments, and those are called Okazaki fragments, that are then going to be joined by an enzyme called DNA ligase. I guess the best way to remember it is ligand something, or it glues it back together. This research actually indicates that DNA application might be more like complex, in which DNA moves through rather than moving along the DNA. The DNA actually moves through this group of enzymes. And I'll show you. So here's a group of enzymes here, and it's actually showing maybe the DNA is what's moving through it instead of the enzymes moving, which is current research. In bacteria, which typically only have one circular DNA, there's only one origin of replication instead of many because it's circular. So again, if it goes in two directions, it'll finish the circle. During DNA replication, DNA polymerase is also going to proofread each nucleotide against the template when it's added. If it finds errors, it's going to remove the nucleotide and resume synthesis. So, for example, if your DNA strand is like this, and it's supposed to have UT, UA, UG, UC, and it accidentally inserts another A here, that's going to be problematic. The DNA polymerase is going to go in and fix that and make it right. Sometimes that doesn't happen. The enzymes don't work properly. Usually, something else later on, another enzyme is going to come in and it's going to fix it. But in some cases, it doesn't, and that can actually lead to cancer if you don't have proper DNA proofreading or repairing mechanisms. We are constantly subjecting our DNA to damage. When you go out in the sun, every time you go out there, you're susceptible, subjecting your skin cells to UV radiation, which is going to damage the DNA in those cells. Most of the time, those errors are corrected before they become permanent. If they become permanent, that's what we call mutations. And those can actually perpetuate and continue to replicate the same errors over and over. And that can lead to problematic things like cancer. Ectodermal dysfunction is actually an inherited defect in one of the nucleotide extrusion repair mechanisms. So it doesn't repair the DNA. Usually these people end up with skin cancer because they can't repair the damage. Once the cells are out there and they get damaged, they can't repair it. And so they're more susceptible to skin cancer earlier in life. Mutations are permanent changes in DNA. If they occur in your sex cells or germ cells, they will be passed on to the next generation. Some mutations can be beneficial, and natural selection can favor those mutations. We're going to talk about this a lot when we get to natural selection. Ultimately, this is what is responsible for new species. So, for example, say that there's a rabbit, and there's a mutation that causes the rabbit to be able to jump a little bit higher. If that mutation is an advantage because it can get away from predators, they will survive better and pass that trait on to their offspring, and that will continue through the population until the population becomes so different that they might even form species. Giraffes. When you look historically at giraffes, the average neck length was shorter, and over time the neck has become longer, and that is because mutations have arose that made the neck longer, and it was actually an advantage in the population because they could reach trees that were a little taller and they could eat more leaves, and they survived better and passed that trait on to their offspring. Because DNA polymerase only adds to the 3' end, there's no way to complete the 5' ends of the daughter strand, which means that repeated replication produces shorter and shorter DNA. I'll show you a picture of that in a minute. Prokaryotes have circular DNA, so that's not the case. DNA molecules have nucleotide sequences at the end called the telomeres, which do not contain genes. Usually they're a buffer that protects the organism's genes during replication because that end is not going to get replicated. Telomerase catalyzes the lengthening of telomeres in germ cells, thus restoring their length and compensating for the shortening. But that enzyme is not active in most body cells. So that's why over time, our telomeres get shorter and shorter. And I'm going to have you do a reading this chapter on how that seems to be linked to aging. Cancer cells often show unusually short telomeres, which makes sense because they divide so fast. The DNA is copied often that they're going to be losing a lot more of those telomeres in a faster amount of time. So this just shows you, here's your RNA primer, and these primers get cut out. And so it gets shorter and shorter over time, and usually it's only a few bases in a long sequence of bases, so it doesn't make a big difference. And again, usually the ends don't have a lot of important genetic information, so that kind of buffers from losing any genetic information. It just shows again closer up picture of that. DNA is packaged into chromosomes contained in the nucleus of eukaryotic cells. DNA in bacteria is in a dense region called the nucleoid, but it's not actually a membrane-bound part. DNA is called chromatin or chromatin in the nucleus prior to mitosis, at which time it coils up into the chromosomes. So here's a double choice DNA. These are called histones, and then the DNA wraps around the histone several times, and together that's called a nucleosome. And then that's how DNA is packaged so tightly into those chromosomes. Because if you stretch out your DNA in a cell, it's like the length of a car, but it's getting into tiny, tiny little cells, and that's because it wraps around these histones and packages really thin. You can see that here into that chromosome. There's a lot of DNA in that chromosome. To kind of extend what we talked about with genetics, maybe right here, the sequence, and stretch that out of DNA, that is a gene, like code for some kind of trait. Right, so I did talk a little bit about RNA primers. RNA is going to play a bigger role in protein synthesis, but RNA is a molecule that's similar to DNA, but there's just enough differences that it's not recognized by the cell as being the same as DNA, so it can leave the nucleus and do other things. DNA is called the nucleus. The DNA is a double strand. RNA is typically some kind of single strand, and there are actually lots of types of RNA, and they all look a little bit different, but none of them are a double helix. DNA has deoxyribose sugar, and RNA has ribose sugar. DNA doesn't leave the nucleus, but RNA does. And DNA contains thymine, which is going to pair with adenine, but RNA does not contain uracil. So A is going to pair with U if we're talking about a strand of RNA. And that's it for these notes. I'm sorry they were so long.